Next topic is about cell-to-cell communication and development. Morphogenesis is the result of cell-to-cell communication through cell signaling pathways in the development of organisms. During morphogenesis, each type of cell in the developing embryo are arranged into distinct patterns to form structures of the tissues and organs because cells have different sets of proteins in its cell membrane. Differential cell affinity. Presumptive epidermal cells, dissociation of cells, neural plate cells, spontaneous reaggregation, segregation of cell types, neural cells, epidermal cells, cross section. The differential cell affinity in the embryo is primarily due to presence of cell adhesion molecule cadherin, structure, function and types. Cadherins are transmembrane proteins having a 5-domain extracellular end that interact with other cadherin homophilic interaction on adjacent cells, forming adherens junction and intracellular end anchored by a complex of proteins called catenins. So cadherins are calcium dependent adhesion molecule. Cadherins are calcium dependent adhesion molecule. Therefore, the second domain of extracellular end have calcium binding site. The cadherin catenin complex bind to the actin cytoskeleton of the cell, thereby providing the mechanical forces for forming tubes. In vertebrate embryos, several major cadherin types have been identified. E. cadherin is expressed on all epithelial tissues of embryo. P. cadherin is found predominantly in the placenta, where it helps the placenta stick to the uterus. N. cadherin becomes highly expressed on the cells of the developing central nervous system. Then R. cadherin is crucial in retina formation. A class of cadherins called protocadherins lack the attachment to the actin skeleton through catenins. The timing of particular developmental events can also depend on cadherin expression. For example, N-cadherins appear in the mesenchymal cells of the developing chick leg before condensation to form nodules of cartilage. If the limbs are injected just prior to condensation with antibodies that block N-cadherin, the mesenchymal cells fail to condense and cartilage fails to form. Then cell migration. The first stage of migration is polarization, where in a cell defines its front and back directed by diffusing signals from ECM that recognize the cytoskeleton. The second stage of migration is protrusion of cell leading edge by polymerization of the actin microfilaments at the cell membrane, creating long parallel bundles. The third stage of migration involves the adhesion of cells to its extracellular substrates. Integrin spans the cell membrane, connecting the ECM outside the cell to an actin cytoskeleton on the inside of the cell to form focal adhesion, hemidesmosomes. Fourth stage, release the adhesion by proteolytic destruction activated by Ca2 plus ions and allow cells to migrate in forward direction. Then cell signaling, introduction and competence. The precise arrangement of corneal and lens tissue in vertebrate eyes formed by coordination between cells, where one group of cells changing the behavior of an adjacent set of cells. There are changes in the mitotic rate. The kind of interaction at close range between two or more cells or tissues are different histories and properties is called induction. The first component of the inductive interaction is the inducer, the tissue that produces signal paracrine factor that change the cellular behavior of other tissue. The second component, the responder, is the tissue being induced. The ability of responder tissue to respond to a specific inductive signal is called competence. Then criteria for performance receptor tissues. Cells of the responding tissue must have a receptor protein for inducing factors, the ability to respond to the signal. The two major parts of the eye construct each other and the eye form reciprocal paracrine interactions. The head ectoderm is the only region capable of responding to the optic vesicle. If the optic vesicle from Xenopus laevis embryo is placed underneath the head ectoderm in a different part of the head from which the frog's optic vesicle normally occurs, the vesicle will induce that ectoderm to form lens tissue. Trunk ectoderm, however, will not respond to the optic vesicle. Only head ectoderm is competent to respond to the signals on the optic vesicle by producing lens. Normal induction by lines in optic vesicle. Optic vesicle cannot induce ectoderm that is not complete competent. Optic vesicle is removed, no lens is induced. Tissue other than optic vesicle is implanted, no induction occurs. Then one induction will give a tissue the competence to respond to another inducer. For example, the first inducer of cells may be the ectoderm and mesoderm that underlie the cells forming ectoderm at mid-gastrula stage. So PAX6 is a transcription factor in the anterior ectoderm and act as competence factor and provide competence to respond to the inducers from the optic cup. Then inducer molecules. The optic vesicles appear to secrete two paracrine factors, a BMP4, a protein received by the lens cells and induce production of SOX2-TF. Then FGF8 signal produces the appearance of LMAF-TF. So in the ectoderm, the combination of these TFs activate lens-specific genes such as delta crystallin. Types of interaction between cells on the basis of source and action of signaling molecules. Autocrine interaction. When the same cells that secrete paracrine factors, it has its own receptor for response to them. Example, placental cytotrophoblast cells synthesized and secrete PDGF, whose receptors is on cytotrophoblast cell membrane, result in explosive proliferation of the tissues. Autocrine signaling molecule. Paracrine interaction. When proteins synthesized by one cell and can diffuse over small distances to induce changes in neighboring cells. Example, vertebrate eye lens induction, ectoderm by optic vesicle cells, paracrine signaling cell, target cell, local mediator. Juxtacrine interactions. When cell membrane proteins on one cell surface interact with receptor proteins on adjacent cell surface. Example, Notch signaling pathway involved in neural development, contact dependent juxtacrine. Signaling cell, target cell, membrane boundary signal molecule. Then endocrine interaction. Cells producing signaling molecules are secreted into the immediate blood vessels and travel through the bloodstream into the target organ and exerts their effects. An example, hormones action through these interaction. An instructive and permissive interaction. An instructive interaction, a signal from the transducing cell, is necessary for initiating new gene expression in the responding cell. Without the inducing cell, the responding cell is not capable of differentiating in the particular fate. Example, when the optic vesicle is experimentally placed under a new region of the head ectoderm and causes that region of the ectoderm to form a lens. If the inducing cell is changed, the differentiating fate of the responding cell is changed according to the signaling molecule. In permissive interaction, the responding tissue is already specified and needs only an environment that allow the expression of these traits. Example, many tissues need a solid substrate obtaining fibronectin and laminin in order to develop. These molecules does not alter the type of cells that is produced, but it enables what has already been determined to be expressed. Then epithelial mesenchymal interactions. All organs consist of an epithelium and an associated mesenchyme. So responding cells specify accordingly from the region it belongs in response in induction. Regional specificity of induction. Skin is composed of two main tissues, an outer epidermis, epithelial tissue derived from ectoderm, and a dermis, a mesenchymal tissue derived from mesoderm. Reciprocal interaction occurs between them through signaling molecules. The dermal mesenchyme plays an instructive role that activate a different set of genes in responding epithelial cells. When cells from different regions of the dermis are recombined with the epidermis, the type of cutaneous structure made by the epidermal epithelium is determined by the original source of the mesenchyme in chick. So responding cells specify according to the genes activate in response to induction. Genetic specificity of induction. Here mesenchyme can instruct the epithelium so as to what set of genes to activate, but the ectoderm responded by making the only kind of mouth it knew how to make, no matter how inappropriate. So organ type specificity. Feather claw is usually controlled by the mesenchyme. Species specificity is usually controlled by the responding epithelium. Then reciprocal transplantation between the perceptive oral ectoderm regions of salamander and frog gastrulation leads to newts with tadpole suckers and tadpole with newt balances.